Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today we're going to be talking about the cell cycle. First, let's talk about how different cells replicate in different ways and some terminology, starting with what a karyotype is. A karyotype refers to the complete set of chromosomes in a cell, including their number and appearance. Ploidy refers to the number of complete sets of chromosomes in a cell. In animals, autosomal cells are diploid, or 2N, which means that they contain 2 copies of each chromosome. Germ cells, on the other hand, are haploid, or just 1N. These contain 1 copy of each chromosome. In humans, these numbers are 46 and 23. We inherit 23 chromosomes from each parent. Also, as a reminder, autosomal cells, which are also known as somatic cells, refers to all the cells in the body except for germ cells, which are sperm and egg cells. This is why germ cells are haploid, as a sperm and an egg get together, so both are haploids and make a diploid cell. The process of this is meiosis. We will talk about meiosis in a later episode. Today, we will talk about mitosis, in which eukaryotic cells replicate through the cell cycle. The cell cycle is a series of tightly regulated steps that a cell goes through to grow, duplicate its contents, and divide. It's essential for replacing damaged cells, allowing organisms to grow, and ensuring that every new cell has exactly what it needs to survive. For actively dividing cells, the cell cycle is traditionally divided into 2 broad stages, interphase and mitotic phase. For cells that are not currently dividing or do not divide, there is a resting phase called G0. Here, the cell is simply living and serving its function without any preparation for division. Cells like neurons, red blood cells, fat cells, and muscle cells always stay in G0 as they do not divide. Other cells only enter G0 phase for a short period of time. G0 is an offshoot of G1 phase. With that, let's talk about interphase. Interphase is where the cell spends about 90% of its time. It's subdivided into the G1, S, and G2 phases. So, G1 phase is also known as gap 1, or the presynthetic gap. This is the first growth phase. In this phase, the cell grows in size, synthesizes mRNA and proteins needed for growth, and the cell accumulates building blocks of chromosomal DNA, as well as sufficient energy reserves to replicate each chromosome. To continue on to the next phase, which is the S phase, or the synthesis phase, the cell must pass through a regulatory checkpoint known as a G1/S checkpoint. This is the main decision and restriction point. Here, the cell checks for DNA damage, proper cell size, and whether the cell has everything it needs to replicate. If it does not have everything, it is not permitted to continue and must grow more. In the S phase, DNA is duplicated. By the end of this phase, every chromosome has been copied and is now composed of 2 sister chromatids that are bound together at the centromere. This ensures that each daughter cell receives a complete set of chromosomes. This phase involves complex processes that take significant time due to the complexity of the genetic material. A note to remember here is that the ploidy of the cell does not change even though the number of chromatids has doubled. So even though 92 chromatids are present, humans only have 46 chromosomes at this stage. The next phase is known as G2, or gap 2, or the postsynthetic gap. In this stage, cells have twice as much DNA as cells in G1 do. In the G2 phase, the cells continue to grow and they prepare for mitosis. This includes things like having enough organelles and having enough cytoplasm. In the G2 phase, there's also a G2 checkpoint. This ensures that DNA replication was completed and no DNA damage was present. If the cell can pass this checkpoint, it will now enter the mitotic phase, and we will go over the mitotic phase and cell cycle regulation next time.